So we've established that DNA makes up chromosomes, and the DNA is responsible for our genetic makeup and sort of helping particular organisms and species to produce things that they need and look and appear and act the way that they are. So we're going to take a look in a bit more depth at the particular structure of DNA and how the arrangement of different parts within the DNA can give rise to different species, different features, and that kind of thing. So one of the most important things to know is that DNA is made up of these structures called nucleotides, and each nucleotide is made up of 3 key parts. So nucleotides are made up of a phosphate. I'll draw a little key on the side here. So we have a phosphate group, often drawn as a circle, and then we have a sugar, which is often drawn as a pentagon, and then I'll just use squares for something called our bases. So nucleotides are made up of 1 phosphate, 1 sugar, and 1 base, and then DNA is made up of several, or more than several, hundreds or thousands or tens of thousands of these nucleotides. So what we see in DNA is firstly we'll have something called our sugar phosphate backbone. So we have a whole bunch of sugars and a whole bunch of phosphates, which are linked together with covalent bonds. Alright, I'll do one more. So here we can see we've got a phosphate, sugar, phosphate, sugar, phosphate, sugar, and so on. Now what we see to each of these sugars will be connected a base. So the easiest way to remember this is that sugars are sticky, so bases stick to them. So I've got a sugar here, and I'll draw in a square. So to each one of these sugars we have a base, and this is essentially one strand of DNA. So one of these phosphate groups, one of these sugars, and one of these bases together, put a big circle around one of them, like so. This would be an example of a nucleotide. So there you can see we have 4 nucleotides in that strand of DNA. So what we also need to know is that there's 4 different types of bases that we can see present in DNA. I'll write that over here. We have 4 bases. These are adenine, guanine, cytosine, and thymine. And we can often, or you often see them represented just with the first letter of their name. So an A, a G, a C, or a T can be used, and we'll know which base we're talking about if we're talking about DNA. So the squares there that are attached to our sugars that make up part of the nucleotide, they could be one of those 4: adenine, guanine, cytosine, or thymine. Alright, I'll just remove some of this stuff to make a bit more room. Now what we see is that not only will we have one of these chains of DNA, or lines of DNA, we will have a second which is going to pair up with it, which gives DNA this double helix structure. We'll talk about it in a sec. So what we'll have is, I'm drawing the bases first, kind of the exact same structure lining up on this other side. There's a bit of a dodgy pentagon. I'll draw in our phosphate groups, and it will look something like this. Now the bases actually face each other, so the bases on the opposing nucleotides on the opposing strands, they are weakly held together with something called a hydrogen bond. So these are hydrogen bonds here, whereas these are more stronger covalent bonds. But the hydrogen bonds are weaker, which is really important for a couple of reasons. One, which is because we can open this DNA double helix up, which is important for things like replication, which we'll look at later. The other point to note about DNA is the fact that these bases will only pair in a particular manner. So these little hydrogen bonds between the bases of each nucleotide are what holds the two strands together. Now, if we have an adenine here, adenine will only pair, because of its structure, only pair with thymine. Okay, same as if we had a thymine on this strand, obviously it works both ways. So adenine and thymine are only two bases that will want to pair with each other. And adenine and guanine and adenine and cytosine, they will not be able to form this attraction. Our other two bases bind together as well. So our guanine and cytosine, so G for guanine, C for cytosine, these are the only two bases that will want to hold onto each other, form hydrogen bonds with each other as well. So we must have that particular arrangement in order to get this DNA to be able to bind together via the bases. Last little point that I want to make, if I scroll down to this picture here, if I can move it into the middle and make it a bit bigger perhaps. There we go. So what we can see here is we've got our chromosome. So the reason why our chromosome is not in a linear shape, it's an X shape, it just means that our chromosome here, the DNA has doubled. So we've got a double linear shape because it's ready for replication, which we'll talk about a little bit later on. So it's a linear chromosome, but just with a double copy at the moment. And what we can see is that DNA will make up this chromosome, and the DNA will be wrapped around these little ball type structures here. So these balls are called histones. They're a type of protein, and then clusters of histones can be grouped together to create nucleosomes. Nucleosomes probably not a term that's overly important, but more so histones are the proteins that these DNA structures are wrapped around. So what you can also notice down the bottom here is, I'll just highlight this bit here, is that we've got that ladder type structure where the bases in the middle of a sugar phosphate backbone are holding together, but it's twisted. So we call it a double helix. The reason why we call it a double helix is because there's two strands of DNA that are linked together, but we've given it a big twist. So you imagine you take a rope ladder and you kind of hold the top and the bottom and you twist it up. Also important to note there is we've got an example of a gene. So particular sections of the double helix will make up particular genes. So what that gene does is there's a particular section of DNA that have a particular recipe of how to make something for the cell. It might be a protein that has a function or a structure in the cell. It might be particular proteins and things which can help to give us our hair color or eye color or particular behaviors or something like that. So then what we'll notice is that double helix, you can see from the picture, we've got our DNA there, wraps around those histones grouped together in nucleosomes. So histones, a type of protein, grouped together in nucleosomes, and they all bunch together in order to make our overall chromosome. Remember, chromosomes are linear in eukaryotic cells, so animals and plants, and circular in prokaryotes like bacteria.